Okay, so light excites the electrons, and as it enters the PS2, which is the blue funnel here, and then the two marbles will fall down through here, and they're representing H plus and O2, and then the electrons move through the transport chain, creating O2, and it places an additional H plus in the lumen, which is represented by this container here, and then it starts the ATP synthesis. And meanwhile, the one over here, it goes through the cytochrome complex through this, it's the wheel, and then comes again through the PS1, and then more light excites another pair of electrons and creating an ADPH.
So 6 CO2s enter into the dark cycle, which we have represented here. And as they go in, ribulose biphosphate is reduced. And when it's reduced, it uses 6 ATPs to do this, which is represented with this wheel. And after that, it gives off 6 ADPs from the byproducts of ATP. Then the NADPH that exits the light cycle comes into the dark cycle, carrying its electron, and drops its electron off. And NADP plus and phosphate leave. And as it goes down, it goes through, and glucose exits the cycle, and the cycle continues to go back to form ribulose biphosphate again, and it's done through Rubisco.
Alright, we take that glucose and it goes through here, the marble goes here, and breaks off into a phosphate that joins onto the glucose from the cytoplasm using ATP right here. This makes glucose 6-phosphate. Glucose 6-phosphate is then rearranged by an enzyme, which we show right here, to form fructose 6-phosphate. There you go. Then a second phosphate is added using ATP to form fructose 1,6-biphosphate. Next we have the cleavage phase, which is represented right here, which breaks 6 carbon molecules into 2 glyceraldehyde 3-phosphate, which we have shown here, breaks off into 2. Lastly is the liberation phase, and it produces 4 ATP, 2 NADH, and 2 molecules of pyruvate, which is going to be represented by these 2 cars that will shoot off.
Okay, so this portion here is a citric acid cycle. So it starts when our marble comes up and it attaches here. So then when the marble attaches, it represents the acetyl CoA attaching to the oxalacetate to form citrate. And then as the marble moves around this curved track here, this citrate is rearranged to the isomer isocitrate. And then once the marble drops into this purple basket here, the isocitrate is oxidized to an alpha ketoglycerate. Here the CO2 is released and NODH is formed, which is shown also by the many triangles there. And then when it drops to the next level through the basket, the alpha ketoglycerate is oxidized to combine with CoA to form succinate-CoA. CO2 is broken down to CoA and succinate. Here, the succinate is oxidized to fumarate. FADH is made when the marble passes through this bridge right here. And then when the marble rings this next spell, it signifies the fumarate combining with water, and malate is created. And then once the marble releases to the very bottom, the malate is oxidized to oxaloacetate, NADH is made again, and the cycle begins here. Restarts.
Okay, so this is the final process of cellular respiration, and it's the synthesis of ATP via oxidative phosphorylation. This process is taking place in the inner membrane of the mitochondria. There are 4 complexes, which is this, and this, represented throughout by various pieces of toys. NADH and FADH2 are symbolized by a marble, which is at the bottom. NADH enters complex 1, which is represented by the yellow track that carries the marble. As electrons are passed down the chain, energy pumps hydrogen ions across the membrane. This established an electrochemical gradient. NADH travels directly to the complex 3, which is this. which is the green bridge. NADH2 enters at complex 2 when the marble travels through the red piece, setting the bell off, then continues to travel to complex 6. Just like that. At this point, FADH2 and NADH are now represented together by the one marble. FADH2 and NADH move to complex 4 as seen by the black hole that drops the marble. The electrons then reach the end of the chain, which is the purple funnel. The electrons are ultimately transferred to oxygen, the terminal electron acceptor, which converts the oxygen to metabolic water. The white tube at the bottom of the funnel represents the enzyme ATP synthase, which utilizes the energy from electrochemical gradient of hydrogen ion synthase ATP. And that is all this. That's it.